Well, thank you for that introduction. And really, I'm delighted to be able to share with you some of the humanized mouse work that our group has been a part of over the years. The title of the presentation is really a brief follow-up study to some other work. And in order to frame those findings and give a little bit more context and rationale for why we had approached that, today I'm going to give you an overview, which really is a history of the studies that we have done previously that I think will provide a little bit more clarity. So when I had just started my postdoctoral fellowship at Harvard in 2010, I was eager and looking for a project to work on, and I had come across some studies that I was really fascinated by that came out of just a lot of these individuals listed here, and Mike Brem as well, just demonstrating this utility of immune humanized mice as a way to study human immunobiology. And so, as was mentioned, my training as a PhD student was really in the IBD space, and I had made that transition to trying to understand mucosal immune aspects of IBD in my postdoc. And I thought this would be a really interesting way to try to build upon the murine models that we had been using, and nobody had been doing it at that time in the IBD space. And so I thought, well, this is really interesting. And so while a lot of information has been learned from genetic models, they've taught us a great deal about the specific proteins involved in an inflammatory response. There are cases in which the mouse does not fully mimic or predict the response that we see in human subjects. And in kind of pivoting back to the IBD world, one of the reasons that really launched us into getting interested in using immune humanized mice to study IBD really was because the postdoctoral advisor, Scott Snapper, that I was working with, had been involved in trying to define monogenic disorders that were the cause of infantile or very early onset IBD. And so they had identified mutations in the IL-10 receptor and IL-10 as well that were sufficient to cause disease. But the problem really was that these present early in life, usually in children less than 2 years of age. And so the opportunity to obtain samples to study really the mechanisms driving disease pathogenesis is really difficult. And we thought perhaps if we could use immune humanized mice, maybe that would give us an opportunity to kind of study and characterize some of the immune dysfunction that was happening in these patients. And so while very early onset or infantile IBD is often monogenic, in adult populations, it's actually a polygenic disease. And IBD is quite complex, and the etiology is really a combination of genetic susceptibility that tends to lead to exacerbated immune responses to the intestinal microbiota. And there's also this component of environmental triggers. And I would say that while we know that that's plays a role, it's probably the least understood component of the disease. And the reason this is all important is because in the US, there's over 2 million Americans that are living with IBD. And over the past several decades, the prevalence has been increasing, especially in developed nations. And IBD in general can be classified into two major forms, the first being ulcerative colitis and the second being Crohn's disease. And while both of these diseases have some features that are common to both of these subtypes, ulcerative colitis is restricted to the colon and is continuous, whereas in Crohn's disease, this can occur in any part of the digestive system, and the lesions are patchy and often transmural. And so it's very likely that these two categories actually are made up of other subtypes as well that hopefully one day could be stratified into learning how to treat specific patient populations. But I would say what is common to both of these diseases is really, again, this inappropriate immune response or overactive immune response in the intestine. And while this is really an oversimplified representation of the mucosal immune system, what I want to draw your attention to for this talk is really centered on a type of immune cell called a regulatory T cell or Tregs. And these cells are critical regulators of effector immune responses, not just in the intestines, but other parts of the body as well. Now, these cells actually can inhibit effector cell responses through a number of different distinct mechanisms, and that can occur through the secretion of immunosuppressive cytokines, as well as direct cytolysis using granzymes, or metabolic disruption by sequestering IL-2 or ATP hydrolysis. And all of these different mechanisms really allow regulatory T cells to balance the effector T cell responses in these compartments, including the intestine. Now, in most individuals, that happens normal, but in some individuals, this balance, for whatever reason, it could be through a lack of Treg numbers or Treg function, but you can get this shift toward you tilt the balance towards this effector T cell response, which can lead to chronic inflammation and or autoimmunity. Now, in terms of ontogeny, where do these Tregs come from? So I would say most of them can be classified into two different flavors of Tregs, if you will. The first really are ones that originate in the thymus, and these regulatory T cells are selected against or for self-antigen recognition, and they really function in the periphery to limit self-reactive T cell responses. However, another type of regulatory T cell occurs in peripheral lymphoid compartments where they are induced from naive T cell precursors, and these regulatory T cells are mostly involved in tolerance to other foreign antigens, so commensal bacteria or food. And I would say that probably the best way to appreciate the critical nature of Tregs in their role in regulating immune responses can be seen by in patients that have loss of function mutations in the lineage defining transcription factor FOXP3 for regulatory T cells. This leads to autoimmunity, type 1 diabetes. It's often fatal within the first 2 years of life unless those patients receive a bone marrow transplant. And like humans, mice that are deficient for FOXP3 also develop systemic inflammation, autoimmunity, and premature death. Now, given our interest in human IBD, the benefit of employing humanized mice were really attractive since there's no risk to the patient and the environment is a little bit more controlled, depending on the vivarium. But we can also experimentally manipulate them to ask relevant questions pertaining, again, to mechanisms, or in the case that I'm going to show you going forward, assessing different therapeutics. Now, there are different methods for generating immune humanized mice, as I'm sure a lot of you are already familiar with. Probably the simplest is really to take peripheral blood mononuclear cells from subjects and inject them directly into mice. And I'm just going to refer to these mice as NSG mice, since that's what I'm going to be discussing later. These mice are the ones that Jackson makes available. Now, the benefit is that this is a quite easy thing to do. The limitation is time. These mice will develop a fairly rapid xenograft versus host disease, probably about 3 or 4 weeks of age. So one just needs to consider that with the questions that you're asking. The second one that I want to mention is the BLT model, which stands for bone, liver, thymus. This is kind of what is depicted here in this illustration. And it's probably the most robust model in terms of immune responses because the T cells in these animals are selected and mature on a human thymus implanted tissue. But there are several complications with this model. The first being that it requires fetal tissue, which can be complicating. It requires advanced surgical techniques. And also some of these mice will go on to develop a xenograft versus host disease as well. So the model that we actually are going to be using for our studies is kind of somewhere in between. So we take purified CD34 positive hematopoietic stem cells or progenitor cells from cord blood and inject them into newborn pups. And these mice will have pretty good hematopoiesis over their lifetime. And then around 4 months of age, we can kind of use them for different experiments. So as I mentioned, or I didn't actually mention, I failed to mention it, the caveat to that CD34 stem cell model, though, as you'll see here, is really it's been noted that their adaptive immune response is not that great. And we thought this was going to be important because we know in IBD, effector T cell responses are one of the critical factors of driving disease pathogenesis. And so we thought if we're going to use this to study IBD, we probably need to try to figure out ways to improve this further. And so one of the, I think, ideas that we had is really that in a normal mouse, the mouse T cells are selected in the mouse thymus and they go into the periphery and they encounter peptide presented on these mouse MHC molecules where they then can be activated. What happens in a humanized mouse is that you have these human CD4 T cells or CD8 T cells that are selected in the mouse thymus, in the case of the CD34s model, but they're selected in the mouse thymus on a mouse MHC class 2, and if they encounter a human antigen presenting cell, humans express a different form of MHC that's called HLA. And I'm not going to get into the nitty gritty of that, but just so you know that basically you're asking a human T cell to be activated by a cell presenting a peptide on a molecule that it's never really seen before. And so we think that this, at least in part, can explain some of the lack of an effector response. And so really what we aim to do was to kind of ask the question, well, can we restrict or force these human CD4 T cells to be selected on a human version of the MHC class 2? And then when they go in the periphery, perhaps they would have a better, the ability to be activated by human antigen presenting cells would be better. And so this was kind of straightforward, pretty basic stuff. So what we did was we had taken 3 strains that we obtained from Jackson and combined them. And so again, we're using the non-skid gamma, the base strain. Here, we're combining it with this deletion for AB0, which is really just a way to delete murine MHC class 2. And instead, we are, this mouse is expressing human HLA-DR1, which is a class 2 allele. And this class 2 is actually being driven by the mouse MHC class 2 promoter. So it is a transgene, but the expression, as I'm going to show you in a minute, is pretty faithful to what we would expect. And so here on the right, you can just see HLA-DR expression in the thymic epithelial cells here, whereas we don't see that in the NSG, which is expected. So it looks like the expression of this molecule is indeed functioning as we anticipate. And so the first question that we really wanted to ask was, okay, if we humanize these animals, as I mentioned before, using CD34 cells from healthy donors injected into one-day-old pups, one of the easiest ways to assess T cell responses is to do a delay type hypersensitivity reaction. And so what this is we immunize these animals using OVA in an adjuvant, and then 10 days later we challenge them by giving them a secondary recall challenge in the foot pad of OVA, and then we measure how much that foot pad swells. So on the right, what you'll see is that these NSG AB0 mice, these don't have any of the class 2 molecules. They're deficient for mouse class 2 and human class 2. So they really don't have CD4 T cells circulating. You can find a few, but not very many. And as you might expect, they don't have a recall response. The NSG mice have a slightly better recall response, but in our transgenic DR1 expressing mice, you see it's dramatically improved compared to just the NSG. So this suggests that perhaps selecting CD4 T cells on a human HLA molecule actually does enable better T cell activation. Now, we know that T cells can also provide help to B cells. And so we partnered with Wayne Marasco's group at Dana-Farber to really look at whether or not the B cell compartment was improved as well. And so in looking here, this is just kind of assessing the transitional B cells or mature naive cells. And what you can see is that the DR1 expressing mice have fewer transitional B cells and an increase in the mature naive. And so this is, again, suggesting that for whatever reason, perhaps their T cells that are selected on the human HLA are better at providing help to B cells. And what we find in serum is that there is an improvement, and these mice are unchallenged. So this is just at baseline, that there's an increase in circulating IgG, as well as we even see class switching to IgE, which we rarely ever see in the NSG strain, or certainly not in the ones that lack CD4 T cells. So combined, this suggests that, okay, it looks like if the T cells are restricted to HLA, they are functioning or promoting better adaptive immunity in these animals. The real question is, can we model a human immune-mediated disease that is actually driven by human T cells? And so to do that, we had an opportunity, actually, it was really amazing. This is why working at a great institution provides so many opportunities, because we were able to take hematopoietic stem cells that we had recovered from a bone marrow aspirate of a patient that lacked FOXP3. So as I mentioned earlier, FOXP3 deficiency causes IPEX syndrome, which leads to fatal autoimmunity. And so what we did was we took these IPEX stem cells and we injected them into either the NSG-based strain or our DR1 expressing strain. And really the question was to see if they develop systemic autoimmunity. And so what we saw right away was that the DR1 expressing mice receiving the IPEX cells had an increase in mortality in starting around 12 weeks of age, which is really kind of lines up nicely with what others have shown in terms of um, T cells being really developing in these animals. It really kind of peaks and is happening at a more robust time during this time frame. What was striking though is that these IPEX cells in the NSG mice, I think we had an N of maybe 15 and we only lost one animal. And so it looks like that there's something to the DR1 expressing strain that is allowing these T cells to maybe cause disease. But really to look at that further, we had to go back to the histology. And so here what I'm showing is just several different histological images from different organs, including lung, liver, small intestine, and colon. And if you take healthy control stem cells and reconstitute these DR1 animals, you see that there's really no appreciable inflammation in any of these compartments. If you look at mice that lack FOXP3, so these are not humanized mice, these are just black 6 mice, you can look that you do see inflammation in the lung, the liver, and I'm sorry if you can't really see this very well, but there's inflammation in the small intestine. Interestingly, not the colon, and that's something we can discuss later. Now, if you take those IPEX stem cells and put them into the NSG mice, again, they look very much like the control DR1. There's no inflammation that we can see in any of these compartments. But I hope what you can see in this last set of images is that the IPEX stem cells in the DR1 expressing animals do evoke an immune response in these different tissues, very similar to what the FOXP3 deficient mice look like. So in this way, it looks like we have a mouse expressing a human HLA promotes better adaptive immune responses that when reconstituted with a patient stem cells that lack FOXP3, we can model this monogenic disorder. And so I think that at least here at this point in time, we were excited about the possibilities of what this would mean. And really getting back to the IBD space, the purpose of what we were wanting to do all along was really to assess new therapeutics. And so in order to make sense of this, what we wanted to do is we knew that IBD resulted from these exacerbated immune responses. And for years, investigators have known that a particular dioxin called TCDD, which is an active component in Agent Orange, that it activates this aryl hydrocarbon receptor, and that if you treat cells with this, you can induce regulatory T cells. But again, due to the toxicity of TCDD, it's not really a viable option as a therapeutic. And so it just so happened that we had worked closely with Francisco Quintana, who had isolated an endogenous AHR ligand called ITE. And so we really just wanted to basically ask the question, can ITE be used to expand regulatory T cells and employed to limit intestinal inflammation by using an experimental model of IBD? So I'll introduce that in a second, but first I just wanted to kind of give you a comparison between ITE, this new endogenous ligand, versus TCDD. And so again, TCDD is the control. We know that it induces regulatory T cells. And so here in the green, you're seeing that ITE upregulates the expression of FOXP3, other Treg associated proteins and transcripts, including IL-10, granzyme B, CD39. So it looks like this could be a viable option compared to TCDD. And the important thing about ITE is that it's non-toxic. So if it did work, potentially it could be used as a therapeutic. So the model that we were going to employ here to assess this really was taking this DR1 strain and injecting human CD4 T cells. And TNBS, again, it is a chemical that induces inflammation through, it's not really known. It had been suspected that this was caused the T cell mediated inflammation. That's actually not entirely true. I do think T cells are involved, but we can discuss that later. But in any case, what we're going to do is we took autologous naive T cells and induced Tregs using ITE, and we had labeled those Tregs with cell trace violet, and we injected them into these animals just prior to giving the TNBS enema, which is going to induce that inflammatory response. And so what we found is that we can recover these cell trace violet labeled in ex vivo induced Treg cells at the experimental endpoint. And we noticed that these mice that received those Tregs could actually presented with less inflammation, colonic inflammation. It wasn't totally absent, but just in terms of comparing it to a PBS control, these mice seem to have limited intestinal inflammation. So this raised the possibility that you could use these animals to explore therapeutics to treat IBD using TMBS as kind of that chemically induced model. There are other models of IBD that we personally have not explored, but I have heard other labs using dextran sulfate sodium, which many of you are probably familiar with as well. So the rest of this talk, I'm going to show you information and data that we obtained using TMBS as a model. So again, ITE, that was kind of the first attempt at looking at a therapeutic that could that was beneficial or effective at inducing regulatory T cells and could be beneficial for treating IBD, but we wanted something more, I would say, closer to a clinically relevant molecule. And for that, around, I think 2012, work from Jerry Ritz's group and others had shown that low-dose IL-2 could be an effective therapeutic to treat certain conditions like graft versus host disease or HCV-induced vasculitis. And the reason that this works, it takes advantage of this property of regulatory T cells in that Tregs express constitutively CD25, which is a component of the IL-2 receptor. And the affinity for IL-2 in this trimeric form is actually much greater than what conventional T cells express. So conventional T cells express this dimeric form that does not have this CD25 component. There are some caveats to that with in humans, but in any case, what we wanted to do is really to try to see if we could use this to treat IBD. And so first what we're going to do is really just verify that the regulatory T cells were more sensitive to IL-2 compared to these conventional T cells. And so we take peripheral blood mononuclear cells and we just stimulate them with different concentrations of IL-2. What you'll find is looking downstream at the phosphorylation of STAT5, which is the IL-2 receptor downstream target, you see that at lower concentrations of IL-2, conventional T cells do not activate STAT5. If you increase this concentration, you can get activation of STAT5, but we don't want to activate the conventional T cells. We want to be selective to only activating the regulatory T cell population. So here you can see very nicely we get activation at lower concentrations of IL-2, and this is just kind of the kinetics here, concentration curve shown this increased affinity for IL-2 in terms of its ability to activate the receptor in regulatory T cells. Okay, so that's great. Well, what about in as a therapeutic? And so in a very similar manner as I introduced earlier, the only difference being here, we're actually using the fully humanized mice. So we didn't want to take ex vivo Tregs. Sometimes that can be complicated. If we could just have a model in which the only manipulation was injecting low-dose IL-2. So we turn back to that CD34 humanized system, waiting 4 months and then sensitizing against TMBS. And what we're going to do is very similar to the graft versus host disease that had already been carried out in humans. We're going to give a very similar regimen of providing very low concentrations of IL-2 daily to these mice and then induce colitis and see what happens. And so again, if we pull Tregs out of these animals, we see, just like we saw for the PBMCs, we see a selective activation of STAT5, specifically in the Treg population compared to the conventional T cells. So everything looks good there. But importantly, if I can just draw your attention to the bottom slide here, while the mice treated with low-dose IL-2 were not free of inflammation, what it did do was limit the total immune response in the colon here, and the colitis score is shown on the right. So this raised the possibility that it doesn't exclude the possibility that IL-2 is acting through murine cells, but in the next round of experiments, what we decided to do is basically repeat this type of experiment where we're giving low-dose IL-2 to mice that do not have any human cells. And so this is just taking non-Hodgkin gamma mice, treating them with PBS or low-dose IL-2, repeating that same assay, and I'm only showing you the weight here, but what you can see is that, number one, you don't need the T cells to get, you don't need the human cells to get the disease, but you absolutely need them for the low-dose IL-2 to have any therapeutic benefit. So it absolutely does require human cells. So what about the Treg compartment? So here, again, this is kind of a busy slide, but what I just want to draw your attention to is that low-dose IL-2 expanded regulatory T cells in peripheral compartments, including the blood and the spleen, especially when you compare it to the PBS or the control groups up here. We did see a moderate increase in the mesenteric lymph node, but what was really striking is that although we are limiting the intestinal inflammation in these mice in the colon, we really expected to see Treg expansion in the colon of these animals, but we did not. So that was kind of an interesting phenomenon. I think what that suggests is that expanding regulatory T cells in the periphery can actually influence or limit the amount of inflammation and recruitment of cells to a site of inflammation. Where that's happening, we don't really know, perhaps the mesenteric lymph node, but in any case, that's work that's still being teased apart. So I think at this point, really before I finish and we can take questions, what I've set up here so far has really shown that the HLA expression and restriction of human T cells was important for improving adaptive immune responses. It was required for modeling a human monogenic disorder. And here we're using different therapeutics in these HLA mice to show that you can expand regulatory T cells to treat or prevent inflammation in a model of inflammatory bowel disease. But these HLA mice, although I know that Jackson has some of them available, they're not widely available in terms of making them humanized just off the shelf. And really, we wanted to know, and this came out of questions that we had with different industry partners, was, do you need that HLA restriction? Because if you do, then it means you have to use those HLA transgenic animals to be able to assess these types of therapeutics. But what if you didn't? And so we know that regulatory T cells do have antigen-specific functions to limit some effector responses, but there's also bystander effects. So we went back to using this model just in the standard NSG-based strain. So there's no HLA restriction of human T cells. And we just wanted to know, are these a viable alternative to those HLA expressing mice? So using a similar regimen that we did before, what you can see is that while the improvement in disease score is great compared to just the PBS control, again, these animals still do get disease. So it's kind of somewhere in the middle between the HLA expressing mice and these here. But what we found was that in the NSG animals, you don't need HLA restriction to expand human Tregs in the periphery, as you can see in the blood and spleen. Data that I'm not showing is that we did not see appreciable expansion in the mesenteric lymph node or colon, which is kind of similar to what I showed for the DR1 or the HLA expressing animals. And by histology, we can see that again, very similar. While the low-dose IL-2 does not totally prevent inflammation in the colon, it's improved. And so, and when we looked a little bit more carefully here, we noticed that if you look at the Tregs that actually are present in the colon, and this is by immunohistochemistry, you actually see that the number of Tregs you find in the colon correlates with the severity of inflammation, which I think was surprising. So, or at least not predicted, but it makes sense. So I think what we're finding is that you probably don't want to be assessing the efficacy of your therapeutic by looking at colon Tregs. You need to look some, in the blood or spleen, because if you have inflammation in the colon, you're going to find an increase in Tregs, even if you just have your vehicle control. Okay, so it looks like the humanized mice might be a good model in which to use therapeutics that you can then take into the clinic. So we showed that low-dose IL-2 could be an effective therapeutic for treating IBD in humanized mice. What about in patients? And so this is a very limited snapshot of an ongoing clinical trial that Scott Snapper's group is a part of or leading. And basically, they're giving a similar regimen of low-dose IL-2 to patients with ulcerative colitis for several weeks. And what you can see is that these are 3 different patients, and you see different responses. So patient 1 and patient 3 had a really good Treg expansion in the peripheral blood compartment. And also those 2 patients that had that expansion of regulatory T cells here, as you can see by this curve, what was really interesting is that when you look by histology, again, the ones that had expanded regulatory T cells, so that was patient 1 and patient 3, had a reduced Mayo score with improvement both endoscopically as well as histologically for their ulcerative colitis. So I think the value here is twofold. One, it shows that humanized mice can be a very useful preclinical model to assess a therapeutic that you can then move into the clinic and have similar efficacy. And importantly, if one is interested in assessing a therapeutic where the intent is to expand regulatory T cells, the good news is you can use the off-the-shelf NSG mice that Jackson already humanizes as a rapid way to assess this. You don't have to inject them and wait 4 months like we had done previously. So I think the utility of using these animals for these purposes actually is quite remarkable. So really just to summarize this, replacing the MHC molecules with human HLA certainly improves adaptive immune responses in humanized mice. And I think that's important because depending on what your question is or what your goals are, I think this is actually a model that you should use over the standard NSGs. However, if you are trying to assess a therapeutic that can expand regulatory T cells, you don't necessarily need to do that. Here we've shown that low-dose IL-2 certainly selectively expands human Tregs, and that could be an effective strategy to induce remission in patients with IBD. And so those studies are ongoing and hopefully going to be published soon. But most importantly, and the reason we're having these webinars and journal clubs, is that I think we all agree that these mice can be an effective system in which to evaluate therapeutics, not only targeting Tregs, but just for a variety of other purposes that so many of these investigators probably online now have shown in their respective models. So where do we go from here? Well, it's important to know where we've been. Obviously, the SCID mutation was an important mutation to identify in terms of the utility for making these mice immunodeficient. The work by Lenny Schultz and others combining the gamma chain deficiency really provided the platform that I think is the most robust for generating immune humanized mice. And then our group tried to improve this a little bit by eliminating the murine class 2 and instead expressing a human class 2 to improve CD4 T cell responses. We've actually then gone on to develop mice that lack both the class 2 as well as the MHC class 1. This is done by deleting beta 2 microglobulin. And then we've added those transgenes or different transgenes back to that strain now where they express human HLA-1 or class 1 equivalent, and then a human class 2. And then we've actually back crossed. Richard Flavel's done really nice work knocking in certain human cytokines to improve different components of immune cell development, in this case, CSF1. So we back crossed this to the NSG strain and now combined it with all of these different lovely alleles, which are quite complicated, but hopefully the goal is to provide the community with an ever improved resource for making immune humanized mice, but more importantly, to answer the biological questions that you have. And so all of that work came through the dedication from a lot of different people. I really thank Scott Snapper, who gave me just kind of a free rein to pursue this several years ago. And I think it's been not only a fruitful collaboration, an ongoing collaboration, but hopefully can just give something back to and build upon what so many others have done already.